In this middle third of our course, we've centered our discussion around the dual issues of first, how complex multicellular organisms develop, and second, how they achieve the coordination necessary to maintain a favorable internal and external environment. We're going to continue on this theme today, but we're going to turn to a quite different aspect of the problem of maintenance. Specifically, we're going to ask how it is that animals defend themselves from the onslaught of invaders and injuries that are constantly assaulting them, an aspect of physiology referred to as the immune response. In a very real sense, the immune response represents another set of adaptations that permit animals to handle the challenges of a hostile environment. We've already seen, for example, how some kinds of viruses attack bacteria, injecting their own genetic material, and in this way taking over the bacterial cell and converting it into a factory for making more viruses. And we've also seen how bacteria use enzymes called restriction endonucleases to defend themselves. Remember, these are the enzymes that cut DNA at particular nucleotide sequences, making them very useful as a tool in genetic engineering, but also making them a very effective weapon against the naked DNA injected into a bacterial cell by an attacking virus. Multicellular organisms face an even more difficult task in defending themselves because they can be invaded not only by viruses but also by whole bacteria or fungal cells that enter their bodies intact, as well as a multitude of other single-celled and even multicellular parasites and disease-causing agents. And, of course, multicellular organisms being composed of more than one and potentially billions of cells make an especially attractive free lunch for other invading organisms. How then do animals maintain their biological integrity in the face of these kinds of challenges? This question will be the topic of this lecture and the next. Let's start by considering what needs to be accomplished in general. Biological mechanisms that defend an organism from injury and infection generally have to accomplish at least two things. First, they have to be triggered by some stimulus that's produced when an injury or an attack by a pathogen occurs. By the word pathogen, I simply mean any disease-causing agent. In other words, there has to be some mechanism for the organism's system to detect that it's being challenged. Second, there must be some mechanism, or actually more likely a set of mechanisms, that can counteract the damaging effects of the injury or invasion. So in this sense, this is very much like homeostasis. Some feedback signal says that the system has to be adjusted. In this case, the system has to be defended. Most multicellular organisms have some kind of defense mechanism that meet these first two requirements. For example, in a classic set of experiments done on starfish in 1882 by the Russian biologist Eli Mechnikov, he showed that shortly after he pierced the larva of a starfish, that's the juvenile form, with a thorn. He used a tiny form. He found that shortly thereafter, the tip of the thorn that was inside the animal became engulfed with a large number of amoeba-like cells that were attracted to the area. The damaged tissue had apparently sent out a signal indicating that damage had occurred. And this signal had recruited these cells to the area, which then engulfed tiny bits of the thorn in an effort to protect the damaged area. We call this response, by the way, phagocytosis, and we refer to the cells that do this kind of thing in general as phagocytes. Phagocytes simply means eating cells. And we'll see later that phagocytes form a critical part of our own immune response. Indeed, Mechnikov's experiments with starfish led him to be awarded the Nobel Prize in 1908 for his discovery of what's now called the cellular immune response. Now, there's a third ability that would be very useful indeed for defending an organism, and that's the ability to recognize self from non-self. The ability to distinguish between cells and molecules that normally occur in an organism's own body versus foreign material, that is, to distinguish self from non-self, would allow a defense mechanism to go one step further, from a general warning and repair system to a system that can respond at least a little more specifically to a potential threat. To understand the difference here, imagine that a noxious bacteria has successfully invaded the body of some animal. Now, one way to defend against this would be to first identify that there's something wrong in the part of the body where the bacteria has invaded, and then to activate some mechanism that kills everything in that area. For example, the release of some protein-digesting enzymes. This approach would kill the bacteria and prevent infection from spreading. But of course, it would also kill the organism's own cells as well. This would be as though a country defended against an attack against one of its cities by just simply bombing the whole city. You would fend off the attack, but at great cost. As inefficient as this approach sounds, these kinds of non-specific defense mechanisms do play a role in minimizing damage from injury and infection, even in highly elaborate defense systems such as those of vertebrate animals like ourselves. But now imagine that the defense mechanism has this additional ability to recognize self from non-self. In this case, it would be possible to have a mechanism that specifically targeted the foreign bacterial cells for destruction instead of simply wiping out everything in the area. Such a mechanism would be more efficient because the countermeasure could be focused on the invader instead of directed more generally. It's not imagined how this kind of recognition could work based on what we've learned earlier in the course. The defense mechanism simply needs to recognize some aspect of the molecular structure of the offending object or organism, such as the structure of a protein or a glycoprotein on its surface, and to be able to distinguish this molecular structure as one that doesn't ordinarily occur in the organism. The ability of cells to recognize self from non-self also is very widespread in animals. Even animals as simple as sponges can make this discrimination. For example, you can take a sponge and by changing the ionic composition of the seawater it's in and shaking it gently, you can actually cause the cells of that sponge to fall apart. So you now have just a bunch of sponge cells as opposed to an intact sponge. Now, if you take two sponges of the same species, but two individuals, and you dissociate them, as we'd say, the cells in this way, and then mix those cells all together in one jar, then change the ionic composition back so that the cells now start to reassociate, you find that they sort themselves out. The cells of the two individuals come back together, but only with the cells that were originally in the individual they came from. They can recognize self from non-self. All three features of defense systems that I just listed, the ability to detect an injury, the ability to mobilize countermeasures, and the ability to recognize self from non-self are common to many multicellular organisms. However, there are two other properties of defense mechanisms that are characteristic only of the very highly derived immune system possessed by vertebrate animals. The first of these properties is the ability to recognize foreign invaders in a highly specific fashion. In other words, instead of just being able to recognize self from non-self, cells in the organism could also recognize different specific kinds of non-self. In other words, not just lumping all of the foreign material into one category of non-self, but being able to figure out this is this kind of invader, that kind of invader, that kind of invader, and so forth. Now, the second property is the ability to form some sort of memory of a previously encountered pathogen. Specifically, a memory that allows a previously encountered pathogen to be recognized and responded to more quickly. Taken together, the ability to recognize pathogens in a highly selective fashion and the ability to retain some memory of pathogens that have been encountered earlier provide the basis for a defense system that can be targeted with extreme efficiency and great rapidity to counter particular kinds of threats with countermeasures that are specifically effective at dealing with those threats. Now, the fact is the human immune system can do this, as can the immune systems of vertebrates in general, and we've known this for thousands of years. In fact, the Greek historian Thucydides realized this in 430 BC when he wrote an account of the Great Plague that hit Athens at that time. Thucydides, who himself had contracted the plague and recovered from it, noted that people who had recovered from it, like himself, didn't need to worry about catching the disease a second time. And he pointed out that these people could thus serve to nurse the sick. In fact, in the Middle Ages, both Turkish and Chinese physicians routinely would expose people to the dried crusts of smallpox scabs in order to protect the people that they were exposing this way from contracting the disease. Now, if this idea sounds a little bit crude, it turns out that this was commonly used for centuries. In fact, George Washington decided that he needed to do this in 1777 with new recruits to the Continental Army. There was an enormous smallpox outbreak in the North American continent at that time. And as a result, most of the soldiers in his army were dying not from the battles they were in, but instead from smallpox. In fact, in the previous year, he had lost 1,000 men in battle and 10,000 men to smallpox. So he decided to inoculate all new soldiers with material taken from smallpox-infected victims. This was actually quite a controversial and risky thing to do, because the technique for inoculation at that time could often, sometimes, or could at least sometimes, result in a full-blown case of the illness, and in that sense trigger a local epidemic. It was actually a couple of decades after Washington did this, in 1796, when the English physician Edward Jenner had a key insight that provided a safer approach to inoculation. Jenner noticed that milkmaids who had suffered from a milder and non-lethal form of what was called at the time the pox, which we now know as cowpox, presumably caught from the cows that they were milking, these milkmaids rarely, if ever, actually caught the more virulent form of smallpox. Jenner reasoned that the agents responsible for both cowpox and smallpox must be very similar to each other, similar enough that exposure to one would provide protection later on to the other. Based on this hunch, Jenner did an experiment that actually we couldn't possibly imagine doing today. He took a boy and inoculated this boy with cowpox, with fluid taken from cowpox sores. And then after the boy had recovered from a mild case of the cowpox, he then took the same individual and exposed it to fluid taken from smallpox sores. Now, Jenner was hoping this worked. Unfortunately for him in his career, it did. The boy didn't catch smallpox. And very quickly, this new technique for inoculating against smallpox spread through Europe. In fact, to this day, we refer to inoculations like this as vaccinations. And that comes from the Latin root of vacca, which stands for cow, because Jenner had done this with cowpox. The ability of the immune system to recognize specific pathogens and to furthermore develop a memory of earlier exposure to those pathogens, conveying protection from them in the future, are characteristic of what we call the specific immune response of vertebrates. Another name for this is acquired immunity, which contrasts to more non-specific defenses, which are also referred to as innate immunity. Well, what I'm going to do is spend the rest of this lecture talking about these more non-specific defenses or innate immunity, and that will set us up to then turn to the more specific defenses of acquired immunity. So let's turn and look at what non-specific defenses are available to animals to defend themselves. Actually, one of the primary ways that animals defend themselves against pathogens really is to keep them out of their bodies in the first place. The skin, for example, is an incredibly important organ in this regard. In a typical vertebrate, for example, the skin is the largest organ by weight. So, for example, in humans, your skin makes up about 15% of the total weight of your body. What we call skin is actually a very complex layered structure, with the outermost layer being composed of cells that have produced an enormous amount of a structural protein called keratin. This keratin makes these outermost cells particularly tough and resistant to structural damage. Now, these outermost cells are constantly being sloughed off, with new ones developing underneath them and moving outward. And it's as though there's a set of very actively dividing cells, in fact, the most actively dividing cells in your body, that are constantly producing an endless set of cells that are really throwing themselves on the rampart, trying to block the entry into the body of pathogens simply by providing this physical barrier. Now, not only does the skin provide an impenetrable barrier just by being tough, it's also coated with secretions coming from sweat glands and oil glands that make the surface a very inhospitable place for potential pathogenic invaders. For example, the surface of your skin is extremely acidic. It has a pH of only about 3 to 5, which inhibits the growth of many microorganisms. Furthermore, sweat contains an enzyme called lysozyme, which is specifically able to digest bacterial cell walls. Thus, the skin is coated with materials that make it even more difficult for pathogens to live on the outside of it, let alone to get in. Now, along with the skin of a typical animal, there are other surfaces that routinely come into contact with the external environment and thus also provide potential points of entry for pathogens. The digestive tract, the respiratory tract, the urogenital tract, all open to the outside of the body, and so their surfaces also have to be protected. The digestive tract is actually protected in a number of ways. For example, the saliva that you produce contains digestive enzymes, including lysozyme that I mentioned earlier, and in fact is routinely swallowed, taking whatever's in it into your stomach, which is an extraordinarily acidic environment containing hydrochloric acid, as well as a number of powerful protein digestive enzymes. Respiratory tracts and urogenital tracts are typically protected by thick layers of what we call mucus. And mucus is also a component of saliva. Mucus is a secretion that's made up of a protein called mucin. This protein is a very viscous protein that really provides a trap, literally trapping foreign materials and pathogens and providing a matrix for holding them until they die of exposure to, for example, an acidic environment or digestive enzymes. The respiratory tract in particular actually is lined with cells that not only produce mucin, but also cells that have tiny hairs called cilia, which are beating in unison upward, constantly pushing the mucus layer towards the outside where it can be expelled along with whatever it's trapped. Now, as an aside, I should point out that in spite of these adaptations of exposed surfaces of animals' body to create a harsh environment for pathogens to colonize, there are actually many microorganisms, including bacteria and fungi, that actually specialize in living there and do in fact live in and on us. These microorganisms, ones which are at least neutral towards us or perhaps beneficial, are what we refer to as our normal flora. This is from an old term when bacteria and fungus were sort of thrown in with flowers in the great chain of being and referred to as part of the great flora. These normal flora organisms have their own set of adaptations that allow them to live in this harsh environment. And in fact, they are beneficial to us if for no other reason, because by living on our external surfaces, they provide competition for other microorganisms. And in that sense, by competing for space or resources with those organisms, also provide another kind of protection. Well, so it's hard to get into an organism, but it's not impossible. In spite of these kinds of barriers to entry, pathogens often gain entrance into the inside of an organism's body. For example, not all pathogens are deterred by harsh conditions, such as highly acidic environments or the enzyme lysozyme or similar things. The hepatitis A virus, for example, can withstand extreme acidity, including the acidity of the stomach, and thus hepatitis A actually gains entry into the body via the digestive tract when it's ingested. Furthermore, as I'm sure you all know, the skin and other surfaces can be and often are physically damaged. It only takes the smallest cut to provide a potential source of entry into the body for an invading pathogen of some sort. When pathogens do enter the body, they face what is often referred to as a second line of defense, a non-specific set of defenses against injury and infection. Now, in this case, in this sense, non-specific really means non-specific in the sense of not specifically targeted to particular kinds of pathogens, particular kinds of foreign material. It is specific in the sense that generally these mechanisms recognize self from non-self. We refer to these kinds of so-called non-specific defense mechanisms also as innate immunity or the innate immune system. Now, if a pathogen enters in the body, the primary defense against a pathogen, it usually involves phagocytosis by classes of white blood cells that are attracted to the damaged area and begin to eat whatever bacteria or other kinds of foreign material are there. I'm sure you all know that there are many different kinds of cells found in our blood. Invertebrates, so-called red blood cells, are specialized for transporting oxygen, and their bright red color when they're oxygenated both gives the name to these cells and also is why our blood is red when we see it. In addition, there's not one but actually many, many kinds of so-called white blood cells. We refer to white blood cells in general as leukocytes. Different kinds of leukocytes play different roles in defense, and I'll be introducing you to a number of these throughout this lecture and the next as we see what function they play. Now, an important note I want to add here is that leukocytes are not confined to the circulatory system proper, that is, to blood vessels or capillaries. In fact, white blood cells spend much of their time outside the circulatory system, essentially patrolling in what we refer to as the interstitial fluid. This is the fluid that's found in the spaces between cells in tissues. These white blood cells can move in and out of the circulatory system, but they most often return to the circulatory system eventually if they get back there via a set of separate collecting ducts that are generally referred to as the lymphatic system. Now, the role of the lymphatic system is to simply collect fluids and cells that are constantly leaking out of the closed circulatory system. This leakage of blood fluids and cells is actually very important because it allows the blood to more effectively deliver resources to cells, or obviously allows leukocytes to go to spots where they're needed. But what leaks out has to be returned, and the way this works is that lymph ducts collect this fluid, and eventually it gets circulated via what are called lymph nodes, an organ we're going to return to next time, and then eventually enter into a portal near your heart in the circulatory system. Now, with that aside, there are three kinds of white blood cells, or leukocytes, that are especially important for non-specific defense, or the innate immune system. Two of these are phagocytic cells, which means that, as I said, they engulf and digest other cells or foreign material. When damage occurs, a type of leukocyte known as neutrophils are usually the first to arrive and respond. Neutrophils are the most abundant kind of white blood cell. They comprise actually about 60 to 70% of all leukocytes, and they're constantly moving through the tissues of the body, literally looking for trouble. Cells that become damaged, either from physical injury or from assault by pathogens, release chemical signals that act as beacons, attracting these neutrophils to the site. In other words, the damaged cell is essentially sending out a help signal. And this help signal brings the neutrophil in. Once the neutrophil arrives, these cells begin to phagocytose, that is, eat whatever microorganisms or anything else that they don't recognize as being self, much like the amoeboid cells that Metchnikoff described in starfish almost 100 years ago. Now, a second class, important class of phagocytic leukocyte are called the macrophages. Macrophages are recruited to sites of injury or infection more slowly. The reason they arrive more slowly is partly because they're much less common, so there are fewer of them around, and also macrophages need to be turned on, they need to be activated, and chemical signals they receive from the site of damage will turn them on and then bring them into that site. The name macrophage means big eater. These are much larger cells, and so in some ways they're much more effective at phagocytosing any foreign material. More importantly, macrophages are also longer-lived cells. More or less, when neutrophils engulf some foreign material, they usually die shortly thereafter. So they're throwing themselves onto the defensive line, but they don't last very long. Macrophages will live for a much longer time, and furthermore, and really what will become very important as we talk about acquired immunity, macrophages not only engulf foreign material, but they also digest and process it. And macrophages, at least some of them, will eventually make their way to lymphatic organs, such as lymph nodes, where they encounter other kinds of white blood cells and trigger this more specific acquired immune response that we'll talk about later. Now, there's a third important kind of leukocyte that gets recruited in general to a site of injury or infection, which has the particularly graphic name of natural killer cells. Natural killer cells aren't phagocytes. In other words, they don't eat other cells, but instead they kill cells by releasing a set of proteins that are generally called perforins. Now, as the name perforin suggests, what these proteins do is actually attach to the cell that's being killed and create a large hole, perforating the cell membrane, and in so doing causing that cell, the cell that's being attacked, to burst and thus die. Natural killer cells play a specifically important role in recognizing cells that have been invaded by viruses. Actually, natural killer cells also seem to be able to recognize cells of the organism's own body that have begun to develop cellular abnormalities associated with the onset of cancer. And in attacking these precancerous cells, actually seem to provide an important natural defense, at least against some forms of cancer. Now, bear in mind that all of these defensive leukocytes, white blood cells, are recruited by really general signals produced by pathogens or damaged cells, and they attack any material they encounter that differs from normal body cells. Now, in addition to this cellular response, there are also a variety of proteins circulating in blood and lymph and found in the interstitial fluid that also play important roles in nonspecific immune function. One very important class of proteins are the so-called complement system. This includes about 20 different proteins that act in concert to attack the cell walls of invading bacteria and fungal cells. The complement system actually kills cells in a way that's similar to what we saw for natural killer cells. A complement protein or several proteins will bind to the cell wall of a cell recognized as being non-self, and this will recruit a number of other complement proteins in a sequence that essentially also again build a hole in the cell wall, causing the cell being attacked to burst open and die. Now, there's another set of proteins that play another kind of important role in response to viral attack, and these are proteins that we generically call the interferons. Interferons are produced by cells that have become infected with a virus. The production of interferons actually doesn't help the cell that's been infected in any way. That cell is lost. But the interferons are signals sent to adjacent cells, telling those adjacent cells, in essence, that a viral attack is underway and causing those adjacent cells to themselves now start changing their own cellular machinery so that it's harder for viruses to attack them, essentially changing the machinery that we've talked about much earlier in the course associated with DNA replication and transcription. In this sense, the molecular message of interferons is basically, I'm under attack, save yourselves, to other cells around. Now, the recruitment of leukocytes, along with the activation of complement proteins and other proteins, are coordinated as part of a physiological response that we call inflammation. Inflammation is something we talked about earlier when we were talking about hormones, and I referred to how histamine acts as a local regulator that, when damage occurs, causes blood cells around it blood vessels around it to expand, that is vasodilate, and to become more leaky, bringing more blood to the area and allowing the blood and blood cells to leak out more quickly. This response is why an infected area becomes swollen, becomes red, and in fact becomes hot, because there's more blood flow to it. Now, in addition to local inflammation, a more extensive infection can also trigger a systemic, body-wide elevation in body temperature. And this is an interesting thing for us to consider. We call this kind of body temperature change in response to an infection or damage a fever. And it's also triggered by chemical signaling that occurs as part of the inflammation response in a wound or infection. Now, remember macrophages. They're one of those phagocytic leukocytes we talked about, the larger type that's recruited more slowly. When macrophages encounter damaged cells, in addition to damaged cells or pathogens, in addition to starting to engulf foreign material, they also send out a signal, or actually a set of molecular signals that are in general called pyrogens, basically fire-inducing chemicals. These pyrogens are taken up in the circulatory system and circulate throughout the body, like a hormone might, but they have as a specific target cells those cells in the hypothalamus that are serving as thermostats for thermoregulation. Remember, when we talked about thermoregulation, I pointed out that there are particular cells in the hypothalamus of the brain of mammals that are what control the set point for body temperature. Well, these pyrogens basically tell those cells to raise the set point. And then the hypothalamus turns on whatever effectors are necessary to raise body temperature overall, which is thought to be adaptive because the increased body temperature increases circulation, it increases the effectiveness of macrophages and neutrophils, it increases the rapidity with which complement proteins can do their job, and in fact, increased temperature also inhibits growth of some microorganisms. Now, fever provides an interesting example of what I mean when I say all of these responses are non-specific. If you have a fever, your entire body becomes hot, not just the area that's infected. This may be good for helping the infected area, but it actually also may be harmful because if you have too high a fever, or if you sustain a high fever for too long, you actually can be damaged. Actually, you're damaged partly because the very high temperature can denature proteins in your cells. On a more local scale, the same can be said of other nonspecific immune responses we've discussed so far. If these responses are mobilized to too great an extent, then actually more damage can be done than help is incurred. So what happens then if an infection is too large or too persistent or of a nature that can't be handled by these nonspecific defenses on a local level. This is where the more specifically targeted and more efficient defense mechanisms associated with acquired immunity come into play. And these are the mechanisms that we'll talk about next time.